Hi, I'm Elaine Vickers. Welcome to my video series on cancer treatments. In this series, I'm explaining how different types of cancer treatment work. This video is all about chemotherapy. Let's start with what chemotherapy is. Now, the name itself isn't very helpful. It just means chemical treatment. Even cytotoxic just means it kills living cells. What scientists generally agree on is that chemotherapy refers to a large group of treatments, small chemical compounds, typically made of carbons, hydrogens, oxygens, and a few nitrogens, and maybe one or two other atoms. These drugs have been used to treat cancer since the 1940s and 50s. They're especially toxic to cells that are trying to multiply, often by interfering with the cell cycle, the very orderly process that our cells go through when they multiply. You'll often see the cell cycle shown like this. G0 is when the cell isn't dividing, and most of our cells aren't multiplying and they spend most of their time here. When the cell decides now is the time to multiply, it starts to grow, and this is the G1 phase of the cell cycle. In S phase, it makes an extra set of chromosome. Then it grows some more, G2, and it starts making proteins that create the spindle, a network of fibers that the duplicated chromosomes are attached to. Finally, the duplicated chromosomes are pulled apart and the cell splits in two, and this is mitosis. Chemotherapies disrupt the cell cycle in various ways. Some interfere with the copying of the cell's chromosomes, like 5FU and capecitabine. Some prevent the cell from unraveling and manipulating its chromosomes, like etoposide and irinotecan. Some damage DNA directly, like oxaliplatin and temozolomide, and some interfere with the spindle, such as vincristine and paclitaxel. By the way, if you do like my teaching style and want some education for your team, then do get in touch, and there are some links in the description below. Now, it's kind of useful to know what the drugs do, but that doesn't explain why chemotherapy drugs are more likely to kill cancer cells than healthy ones. The usual explanation is that cancer cells divide more often than healthy cells, and so they're more affected by the chemotherapy. But there are a couple of problems with this explanation. One problem is that some slow-growing cancers, like chronic lymphocytic leukemia, respond well to chemotherapy. But some fast-growing cancers are pretty insensitive to chemotherapy. Also, some cancers that respond to chemotherapy to begin with become faster growing when they relapse later. Many scientists now think that what determines a cell's vulnerability to chemotherapy is whether it wants to live or it wants to die. Healthy cells, not surprisingly, usually want to live. Their DNA is intact, their environment is supportive, and when they're damaged, they repair themselves. When they're exposed to chemotherapy, they become damaged, but they recover. Cancer cells, on the other hand, are already damaged and stressed. Their DNA is faulty, their neighbours are telling them to die, and they're living in a toxic and low oxygen environment. They're like a tree teetering on the edge of a cliff. Chemotherapy damages them just enough to push them over. Chemotherapy is by no means a perfect treatment. As you give the patient a higher dose of treatment, more and more of the person's cancer cells are killed. Ideally, you would give enough of the chemotherapy to kill all the cancer cells. But as you give more chemotherapy, healthy cells are gradually getting killed too, especially ones that multiply often, like those in the lining of the gut, those in the hair follicles, and white blood cells. So the dose you'd like to give is too toxic for the patient to cope with. Instead, you give a lower dose, one that is high enough to be helpful, to kill lots of cancer cells, but not so high as to make life unbearable. The narrow range of doses that achieve this balance is often described as chemotherapy's narrow therapeutic window. To recap, the term chemotherapy refers to a large number of drug treatments for cancer. They are small chemical compounds that are toxic to multiplying cells. They kill cancer cells because they are often already teetering on the brink of death, and chemotherapy pushes them over the edge. But these drugs are toxic to healthy cells too, and that's why they cause side effects. Don't forget, if you like my teaching style and you want some education for your team, find me on LinkedIn or contact me through my website. There are links in the description below.